Welcome everyone. Anna here. We're going to go over part 11 of the neurophysiology chapter, and we're going to focus in on neurotransmitters next. Oh my goodness, there is so much we can talk about with neurotransmitters, and I am going to focus on getting this as simple as possible. Okay, so some things that you need to remember is that there are around 50 different neurotransmitters available in the body, okay? And they're not always classically what you think of as a neurotransmitter, okay? It can be weird things like nitric oxide that will act as a neurotransmitter. So the idea here is neuro and transmitter. It transmits between neurons, so between a neuron and a neuron, or a neuron and another structure. So it doesn't always have to be a classic chemical. As long as it's transmitting, it can be thrown into the category of a neurotransmitter. Now, our neurons are not a one-trick pony, okay? Typically 2+ neurotransmitters are being made and used by each neuron, all right? So it's not just one neuron that goes with each one, okay? In terms of the ones that we've already seen, you've got acetylcholine, okay? And you're going to be trying to remember that you're going to use this with your skeletal muscles. You use it with some of your autonomic neurons that we're going to look at later. It can be really important in the central nervous system as well, okay? And then you've got another category. You've got what we call the amines or biogenic amines. Sometimes these are also called monoamines. Okay, put some bullets in here so it's easier to see what I'm doing. Okay, so examples of these would be like dopamine, epinephrine, norepinephrine, serotonin, histamines, among others, okay? So these are going to be particularly important in your brain. Not that they can't be found elsewhere, but they are very important in your brain, and they're very important. What word did I write there? In the brain. And they're really important for regulating the things that are going on in the brain, whether it's behaviors, your biological clock, so forth. So for example, if you get too much dopamine, you will get schizophrenia. Okay, so it's important for regulating behaviors in those areas. But if you get too little dopamine, you're going to get Parkinson's disease. Okay, so important for regulation of various things. All right, another category of neurotransmitters that are particularly important are amino acids. And you've seen amino acids before. We know that we use amino acids to make proteins, okay? But they can also be used as a neurotransmitter. Now, we're just going to look at this in the central nervous system, okay? Some great examples of amino acids that we use as neurotransmitters are glutamate, GABA, and aspartate. Okay. What else? Neuropeptides, which is just a variation on your amino acids because you're dealing with a peptide. So it's a string of amino acids. These are particularly important for dealing with modulation of pain. Some good examples of this would be those lovely endorphins. You've got somatostatins, enkephalins. If you are taking patho, you should be learning about substance P that we use with pain modulation, okay? And then you got the weirdos. We can call them kindly novel or just weird. We're just going to call them weirdos. So novel messengers. So this one's weird. ATP. We know of ATP because we use it for energy, but you can also use it to get fast transmission of signals between receptors. Okay, another one that's weird, nitric oxide. particularly important in learning and memory. Also, carbon monoxide can be used, and ADP. So again, looking up here, the key concept with a neurotransmitter is whatever you're using, its job is to communicate between the neurons, okay? So it doesn't have to be a classic peptide or other type of chemical. It can be weird things like nitric oxide, all right? Let's start looking at some, actually, let's see. No, I think I've got some more notes I want to go over. Functional classes. We just did this in the, ooh, that wasn't supposed to happen. In the previous slide, not the previous slide, the previous presentation, we talked about this already. So you've got this whole thing of you can excite or you can inhibit, okay? So you need to remember that these things do they do these things, excite or inhibit. One of the key things to remember is that a neurotransmitter can do one, the other, or both, okay? What effect it has depends on the receptor and target organ, okay? So some examples, endorphins. All right, endorphins, one of their jobs is to inhibit signals, okay? And it does this by triggering the release of natural opiates, which helps to relieve the sensation of pain, okay? Another one that's really interesting is acetylcholine, because acetylcholine will excite skeletal muscle, okay? But it can excite or inhibit visceral effectors, okay? So it depends on the target organ and the receptor that's present. And it just inhibits cardiac muscles as part of the autonomic nervous system stimulus, okay, which is kind of interesting. All right, so let's kind of flip through a couple of pictures and see if we can get a handle on this. All right, right here we've got an example of an excitatory cholinergic synapse, okay? Now I want you to look over here at this word acetylcholine, all right, and look at this cholinergic. What this is telling you is that you've got a synapse that only goes with acetylcholine. And we call this a cholinergic synapse. We call these cholinergic receptors. So acetylcholine is going to open the postsynaptic receptor. Sodium and potassium are going to move. If it reaches threshold, it'll generate an action potential. This is your classic neuromuscular synapse. So if you're looking over here, you can see the presynaptic neuron. You can see the postsynaptic sarcolemma. And you've got the classic action potential. Calcium comes in, triggers release acetylcholine. Acetylcholine comes over here, goes to the acetylcholine chemical gate, opens it, and now potassium goes out, sodium goes in, and that causes depolarization. And if it reaches over here, you will get an action potential if it gets over here to where the voltage gates are. One thing I like about this particular picture is it does show the acetylcholinesterase breaking down the green acetylcholine, and the acetate goes off here, and the choline comes here and gets recycled back into the presynaptic neuron, which is kind of a nice picture. That's been left off on some of the other ones we've looked at. All right, let's look at the next picture. All right, as you can see, this particular picture did not import well, but that's okay. You can go back to the original PowerPoints and get the notes if you want them, okay? Now here, what we're dealing with is an inhibitory GABAergic synapse, okay? So we're using GABA as our neurotransmitter, and it's going to inhibit. So if we come over here, we see our presynaptic neuron, okay? An action potential comes down here. It triggers the release of GABA into the synaptic cleft. Now, GABA is not opening the same gates that acetylcholine is doing. It's opening up gates that allow chloride ions to move in here, okay? And then the chloride ions are going to go in here, and chloride ions are negative, so they are going to hyperpolarize the membrane, which ultimately leads to the fact that it inhibits that membrane, okay, which is kind of cool. So you're less likely to get a action potential generating. All right, let's look at another one. All right, right here, we are looking at an excitatory adrenergic synapse. Now, another word for epinephrine, an older word, a word that I don't want you to use in this class, is adrenaline. Okay, so adrenergic comes from the first part of adrenaline. Okay, so again, epinephrine and norepinephrine can be inhibitory or excitatory. In this case, we are using an excitatory example, and we're going to use the norepinephrine form instead of the epinephrine form. Okay, so with this one, it's a little bit different, and the reason it's different is right here. Instead of having a transmembrane protein that basically acts as a gate with a tunnel going through it, what you have instead is this transmembrane protein that does not have a tunnel through it. It's just a little protein, but when the norepinephrine binds to it, it changes the shape, and this little section pops off, and it comes over here to another molecule, and then that molecule is going to trigger a whole bunch of little chemical reactions, which then leads to a ligand gate opening, and then you get sodium coming in, which is really complicated. There's so many steps involved, but it's still really, really cool. So because there are so many steps, it is going to be slower than your typical cholinergic receptor or the GABAergic receptors, because it's got to go through this whole mess before you actually get a gate open to allow something in. Okay? But it's still really neat when you look at how it works. All right, let's look at, we got another picture to look at. All right, so I actually already mentioned this when we went over the cholinergic receptor, but we've got a slide that kind of walks you through, the explanations with some notes. So we're just coming over here to 6, where it's showing that the acetylcholinesterase is going to begin breaking down the structures. Now, things that are happening at the same time is neurotransmitters are popping off. Some of these neurotransmitters that are here are going to bind to acetylcholinesterase, and then some of this acetylcholine is going to be binding to the acetylcholinesterase at the same time. Okay, so you got all of these steps co-occurring. Now, in terms of the cleanup effect, you've got different techniques. You can do this classic right here, where you split up the original neurotransmitter, and the parts might all get resorbed, or some of them might float off into the interstitium. Okay, so those are the two basic pathways that are going to take. If it does go out here to the interstitium, usually it's going to get resorbed by a glial cell and somehow return to the presynaptic neuron for recycling. So basically, this part is all about recycling the components to be reused to make more neurotransmitters. All right, next slide. All right, let's give a little nod to neuromodulators, okay? So I want you to look at the word modulate, okay? This is where you basically tinker with degree of expression, okay? So it can reduce the expression or enhance, okay? So alter, change, enhance, reduce. You're just basically going to be changing the rate of neurotransmitters, their ability to either make more, release them, recycle them, break them down, or you can make the postsynaptic membrane more or less sensitive. So one of the most, no, I don't want to like judge how important these things are. One you're going to see talked about in pathophysiology is going to be nitric oxide. It's a pretty simple one. And it basically is going to activate little pathways inside the cell that modulates the perception of pain or something else, okay? One that you might find kind of fun are these endocannabinoids that you are finding in CBD and THC products, okay? These are naturally occurring. The reason these herbs affect us is because we have receptors that match chemicals in those plants, okay? We've got right over here, this is a fun little picture. So I've got a normal synapse, and then I'm showing a synapse where they've taken THC molecules, and you can see that they are binding with the CBD. So right here, these are natural endocannabinoids, and then you can see if I put a whole bunch of THC in here, it blocks these receptors so that the natural endocannabinoids are not communicating with this presynaptic neuron. And that is going to result in altering memory, appetite, pregnancy mood, pain perception, etc. Okay? Enkephalins and endorphins are also nice examples of neuromodulators that we use for inhibiting pain signals. All right? All right, so we're going to finish up with 2 slides where we talk about glutamate, and this really helps to kind of support what you're doing in pathophysiology if you are taking that course. All right, before we look at a picture, let's do some notes on what glutamate is and how it's interacting with the glutamatergic synapse, okay? So the things you want to remember is that it is the primary excitatory neurotransmitter. I'm going to abbreviate that NT just because now you know my handwriting. It's the primary excitatory neurotransmitter in the central nervous system. So that's your brain and spinal cord, okay? Now, glutamate can be dangerous though. So it is tightly regulated so that you do not have a ton floating around in the interstitial fluid, in the extracellular fluid. So you've got low amounts available at any one time. And the reason you have small amounts at any one time is because it is also an excitotoxin. So that if you have too much of it, it actually will start causing cell death. Okay, now with this particular synapse, all right, the glutamatergic synapse, the one that responds to glutamate, you have 2 pathways. You've got two receptors that are particularly important. You have what is called the AMPA receptor, and you have the NMDA receptors. Okay? The AMPA is a chemical gate. The NMDA is a little different in that it is a chemical gate, but it is also, and this is so weird, it's so fun, a voltage gate, okay? With the NMDA, because of this, I need both the glutamate chemical to be present plus depolarization from the opening of the AMPA gate. Both of these are opened by glutamate, okay? But the NMDA also requires a voltage change, okay? Once these things work, all right, calcium ions are going to flow, okay? And they're going to flow through that NMDA gate, okay? And these calcium ions are going to act as second messengers okay? And they're basically going to stimulate or activate various biochemical pathways, which we're not going to study in here, okay? That'll trigger things to happen, okay? Now, we tend to focus really heavily on neurons in this chapter, but this is an example where astrocytes are super important, okay? Because they are going to be the ones that are monitoring and maintaining the amounts of the glutamate in the extracellular fluid. What word is that? It's not the word. So controls glutamate levels. And this is, again, super important because if you have too much glutamate, you will trigger cell death, okay? And we'll look at that on a picture next. All right, so this is a picture that will be in the book that we use for our pathophysiology course. And what you see is we've got a presynaptic neuron over here, and I have the postsynaptic neuron over here. Inside the presynaptic neuron, I have the purple vesicles, which are going to be releasing my glutamate. Okay, so it's releasing glutamate. Right here is my AMPA receptor, and right here is my NMDA receptor. Okay, what is going to happen is the glutamate is going to bind to the AMPA, and that is going to allow green calcium ions to come into the cells. The calcium ions have a positive charge, so it will begin to act as a local depolarization to the membrane. This travels over here. The NMDA gate is both chemical plus voltage. So what you see are these glutamate molecules attaching, but also this is responding to the voltage change. When that voltage change occurs, this opens and I start to get even more calcium ions flooding into the cells. The calcium ions are going to do a lot of different things. Some of them are good, but again, if I get too much calcium, I will get what is called calcium overload, which will cause my mitochondria to dysfunction. I will no longer have sufficient ATP, which will cause the shutdown of the sodium-potassium solute pump. And if you remember with the sodium-potassium solute pump, that is what maintains the osmotic gradient. So now I've got too much water coming into the cell because it's too salty on the inside. The release of the calcium will also cause nitric species, nitrogen species, okay, NOSs, to be released, all right, which is going to start damaging the DNA within the cell, okay. The NOS is going to come out and it's going to be in the form of nitric oxide, which is going to get cleaned up by the astrocyte. Unfortunately, the nitric oxide can trigger this to come into here, and then I've got too much nitric oxide in here, and that will trigger the release of too much glutamate, so that you end up getting a positive feedback loop that causes more and more calcium to enter the cell, causing more and more overload and damage, which is how this will act as an excitotoxin, okay? Excitotoxin, okay? Now, most of the time that doesn't happen, and we maintain a really nice slow cycle between all of these things, and everything goes hunky-dory and no damage occurs. But if it gets out of whack, you will get a positive feedback loop that basically allows way too much calcium and water to enter the cell, ultimately leading to cell death, okay? Which is something you're going to study a little bit more in pathophysiology. All right, that was it for the neurotransmitter video lecture, and I'm going to stop the recording.